DNA replication. Slideshow. Semi-conservative replication. Each parent strand of DNA acts as a template for a new strand. Template. Template strand serves as a model for the DNA strands to be synthesized upon, like the blueprint for the DNA. Origin of replication. Eukaryotic chromosomes are very long, so they have multiple origins of replication, as shown below. Replication forks. After DNA polymerase binds, the DNA molecule unzips and forms two replication forks. DNA polymerase binds to specific sites on the DNA molecule and catalyzes the addition of nucleotides as the new DNA chain grows. DNA polymerase adds nucleotides to the free end of the primer until the replication of that DNA strand is 100% done. RNA primer breaks down, DNA takes its place, and new strand is now 100% DNA. Primer. The short starting strand of RNA. Primer is complementary to the DNA template strand and is synthesized by an enzyme called a primase. Look at that cool picture of primase and primer and polymerase. Yeah. Nucleotides are added in the 3 to 5 direction. The two strands run in the opposite direction, as shown below.
Alex, leave my brother alone.
Thank you for talking during the video, Robin. The phosphodiester bond forms between the phosphate of one nucleotide and sugar of the next nucleotide. This forms the backbone of the DNA. DNA synthesis happens in the same direction the helicase unwrapped it. This is all good until we realize that the DNA is anti-parallel and the DNA polymerase works in the 5 to 3 direction. During DNA synthesis, nucleotides are added to the free end of the new strand because that end has a free hydroxyl group. These nucleotides have 3 phosphate groups. 2 are released in an exothermic reaction, providing energy for the phosphodiester bond. I completely pronounced that correctly. The 5 to 3 direction is all good for the leading strand, which is in the 3 to 5 direction, meaning they all match. To replicate it, all that needs to happen is for the primase to bind to the template, synthesize the RNA primer, and the DNA polymerase to synthesize new DNA. The lagging strand is a whole new scenario. Multiple RNA primers are made, and since DNA polymerase attach in the 5 to 3 direction, the polymers start closer to the helicase. They form the Okazaki fragments, which end up being separated because of the multiple primers. To fill in the gap, DNA ligase joins this fragment. Telomere is repetitive DNA at the end of a chromosome that protects it from deterioration or fusion with other ends. In DNA replication, the telomerase attaches itself to the lagging strand and adds a telomeric repeat sequence to ensure that shortening does not occur. The sequence gets shorter and shorter as you age. And look at this lovely picture of, and this is DNA polymerase right here. These are the template strands, replication fork, goes psh, psh, DNA ligase. These are the Okazaki fragments, right there. This is a leading strand, lagging strand, because lagging, leading. Yep. Memorize this picture and you will live a long and happy life. Thank you for enjoying our slideshow.